Hi everyone, my name is Erik Wright and I'm joining you from the University of Pittsburgh. Today I'll be talking about whether convergent evolution to similar protein structures confounds protein structural search. Now the motivation for this talk actually came from a keynote presentation at last year's ISMB in Montreal, Canada. And after that talk, someone asked the question of whether some fold-seq matches could be the result of convergent evolution, that is analogy, rather than shared ancestry, that is homology. And so you might imagine that you have two structural matches that have some amount of structural similarity, but they have relatively low sequence identity. And so the question is whether these actually share a common ancestor. This is an important question because sharing a common ancestor, homology, is a necessary prerequisite for many downstream applications of structures and sequences. And when I heard this question, I thought it might be answerable. What we really want to do is compute whether the time to most recent common ancestor of these two structures is less than infinity. That is, they are homologous. And we can do that by calculating substitution scores from the structural alignment. And so if something is a homolog, we would expect it to have a high structural substitution score relative to non-homologs, which we can get by permuting the residues in each of the structures. And so using this measure, I applied it to all of the different structural matches in the AlphaFold database using fold-seq. And what I found is that about 2.6% of fold-seq matches lack support. And that lack of support increases as you go to lower structural template modeling TM scores, as you would expect. And so this is the pattern that we would expect, that is, non-homologs, or things that have low support for homology, tend to also have more divergent structures. And in general, people tend to consider strong matches as those that have TM scores greater than 0.5, which is shown here by the vertical line. But what was most interesting to me about this plot is that there are several points at the bottom right. These are structures that are nearly identical, but they have very low support for homology, sometimes zero. And so I investigated this further, and what I found is that there's a statistically significant enrichment for structural repeats in structures at the bottom right. And in fact, 1% of the structures that have high TM score, that is greater than 0.5, actually lack support for homology. And so I wanted to investigate these red dots that represent structures that have structural repeats. And here are a few different examples of what those look like. You can see they're quite diverse, and they have reasonably high TM scores. And if we were going off the TM score alone, we would consider that very statistically significant. But their sequences have relatively low identity based on structural alignments. And so the question is whether some of these structural matches could be the result of convergent evolution or analogy, rather than sharing a common ancestor or homology. And I thought about this further and came up with this hypothesis, that if two structural matches containing repeats were homologous, then they would undergo a process of first repeating, followed by diversification, and then divergence due to, for example, speciation event. That would result in a tree like that at the upper right, which would be from an initial repeat undergoing some diversification process, followed by divergence in two different ancestors, and you can see there are interspersed repeats in that phylogenetic tree. But if these structures were the result of convergent evolution and they did not share a common ancestor, then we would expect a tree more like that at the bottom right, where the two different structures arose independently, then went through a repeat process followed by divergence, and would separate out on a phylogenetic tree. And so I applied this test to many different structures that have repeats. And what I found is consistently there is support for the structures being from different initial ancestors. And so you can see that here. For example, the structures in the top right, you can see from the alignment, which is the color-coded bars on the far right, that these structural repeat units are quite similar to each other, almost identical, but quite diverse or divergent from the other structural repeat units from the other structure. And so you can see this pattern over and over again across many different structural repeats, that there's strong branch support for them having occurred in different ancestors and then therefore being the product of convergent evolution rather than the product of homology. And we can further apply that to even more complex structures. For example, this one shown here, which has a reasonably high TM score, but UniProt gives it two different functions, even though it looks structurally similar. And what we see is that there is strong branch support for a separation of a clade containing the repeats of one structure from the others. I believe this is a problem that contaminates structural repeats throughout fold-seek matches and probably other structural matches. And that these programs should work hard to try to remove this issue in the same way that sequence-level programs mask structural repeats. The problem might be even deeper, though, because we know from sequence-level homology matching that there are other sorts of artifacts that arise from, for example, low-complexity regions that so far are not strongly considered in structural match programs to my knowledge. And so I think this might be a deeper problem than people anticipate currently. With that, I'd like to acknowledge my lab and the NIAID for funding. And if you'd like to learn more, you can scan this QR code to take you to the paper. Thank you.